Good afternoon, everyone. I am Paula Dinnerstein. I am the president of the board of directors of Beyond Pesticides, which is presenting this webinar today. This is the second session of the 42nd National Pesticide Forum. This year, our theme is the pesticide threat to environmental health, advancing holistic solutions aligned with nature. We have several amazing speakers who are in the forefront of research on the effect of pesticides and other chemical toxins on human health. Um, each one will give a presentation. There'll be a couple of opportunities for questions and answers that you'll be directed how to join in. So I'm going to now turn it over to Jay Feldman, the Executive Director of Beyond Pesticides, who will give a further introduction to the seminar and to each of the speakers. Thank you.
Thank you, Paula. And again, welcome all to the second session of the National Forum, the Pesticide Threat to Environmental Health, Advancing Holistic Solutions Aligned with Nature. We talk about improvement in public health and environmental protection as requiring science, policy, and action. We need all three pillars, with the central pillar science informing action and changes in practices and policy. When we launched the Pesticide-Induced Diseases database at Beyond Pesticides over 15 years ago, we began cataloging the body of science that links pesticide exposure to existential adverse health effects for the purpose of empowering informed discussion and advocacy where we live and work. Currently, there are 2,800 independent peer-reviewed studies in the database, divided into 14 categories, 70 subcategories of illness and disease, including cancer, birth defects, brain and nervous system disorders, immune system degradation, respiratory disease, sexual and reproductive dysfunction, and so on. Biodiversity health is included as a category because of its essentiality to life. What becomes exquisitely clear from the database is that the range of adverse health effects are not prevented with the restrictions of one pesticide or pesticide family, but rather requires that we address a constellation of toxic pesticides for which there is a systemic societal dependency. The good news is that these are chemicals that empirical scientific data find are not necessary to grow our food or manage our homes and landscapes. While all the debilitating and deadly illnesses linked to pesticides in the scientific literature are unacceptable and must be central to the public discourse, cancer as a leading cause of illness and death worldwide continues to attack our loved ones in unspeakable numbers, causing wrenching pain for families and friends. Of course, there are challenges in deciphering all the contributory factors to environmentally induced diseases because we live in a polluted world, exposed to chemical mixtures. The scientists at today's forum are cutting through the noise of pollution and connecting the dots to pesticides and other toxic chemicals. The goal is to tap into this knowledge, not with all the technical aspects of the studies that you'll learn about today, but to extract the findings, point to the data, illustrate the preponderance of evidence, and offer preventive solutions. We are dealing with complex interactions for thousands of chemicals that are not fully studied for their cumulative, aggregate, and synergistic effects, and for their impact on vulnerable and high at-risk population groups. Our focus today is breast cancer, pediatric cancer, prostate and testicular cancer, and infertility. The scientific literature just on these health outcomes alone justifies a major shift in decisions associated with any pesticide use, especially given the available alternatives that make petrochemical pesticides obsolete. We're here today about adverse effects to the endocrine system, and yet we rely on endocrine disrupting pesticides found in our air, water, food, on the farm, in our parks and children's playgrounds and backyards and schoolyards. Contaminated sewage sludge, to be discussed, tells a dramatic story of the toxic soup in which we live and spread on our farms and landscapes. The problem is going to be solved from the ground up, from wherever we are now sitting, where decisions are made to use pesticides, pesticides which are not necessary for productive and profitable food production, aesthetic landscapes, and quality of life. For managing land in sync with nature, see the recording of session 1 of this forum on the Beyond Pesticides website. The esteemed scientists speaking today bring us the facts. We are so grateful for their cutting-edge work and benefit from having them with us from around the globe. It is up to us to use these facts to elevate our advocacy for an urgent transition to practices and products that preserve health. With that, a little housekeeping. Please use the Q&A section at the bottom of your screen to place questions that the presenters can address during the discussion sections in the program. And then later in the program, Dr. Panice's presentation will be simultaneously translated into English from her native Portuguese. In the interpretation lab at the tab at the bottom of your screen, it's marked by a globe icon. Please click English to hear the English translation. And don't hesitate to reach out to our staff via the Q&A box for help if needed. Instructions on all that I've just said are listed in the chat and pinned to the Q&A. So now let me introduce our first speaker, Janone Jensen. Janone Jensen, founder and executive director of Health and the Environment Alliance in Brussels, Belgium, and operating throughout Europe, has been an official member of the World Health Organization's European Environment and Health Governance, representing the health sector since 2000. Ms. Jensen is a founding member of the Global Climate and Health Alliance and serves on its board. She has played a leadership role advocating for a more health protective European Union and national policies on endocrine disrupting chemicals for the EDC Free Europe campaign. In November, HEAL released an incredible report, and we'll be hearing from the author after Janone, entitled Chemical Pollution and Men's Health, a Hidden Crisis in Europe. Welcome, Janone.
Thank you, Jay, for your nice introduction and for inviting the Health and Environment Alliance from Europe to be part of this National Pesticides Forum here today. And welcome everyone from my end online. I'm in France, so it's evening here. And I'm really excited to talk a little bit about HEAL. And we can start with the first slide. I think seeing if they are sharing them or not. Yes, the next slide would be great. So I helped set up HEAL about 20 years ago with the vision of helping to create a healthy planet where current and future generations can really benefit from a clean environment to enjoy a long and healthy life. And our mission is to integrate the health protection into environmental policy by informing decision makers about the health benefits of reducing environmental pollution and recommending a course of action for public policy. We're a network. We have over 70 member organizations in our alliance, including both European-wide networks of cancer, asthma, respiratory, or lung charities. We also have not-for-profit health insurers, medical societies, and youth, women, and environmental groups. And we're all working together to bring the independent scientific evidence to European public policies for action. We don't just work on chemicals, we work across environmental issues that impact health, including air quality and climate. Later on, you will hear more about our new report, but first I wanted to share a bit about the frame of our work on pesticides at this webinar. Next slide, please. Our key goal here is to protect people's health by reducing pesticide use in the European Union and calling for a phase out of synthetic pesticides by 2035. Some of our key priorities for our collective work I put here on the slide with our members and partners. First is a roadmap for pesticide phase out, and more recently, a letter signed by over 130 NGOs in Europe to the European Commission President, asking her to strengthen, not weaken, the current EU pesticide laws. You can see some of our campaigns. One that's particularly dear to my heart is showcasing the health impacted groups like farmers and their families. In October, we had a photo exhibition in the European Parliament, and we got to hear very moving testimonies from French farmers and workers from one of our members called Phyto Victims. It's an association that works to get formal recognition and compensation for its members from the French government for occupational exposure to pesticides, which has contributed to their Parkinson's or non-Hodgkin's lymphoma, for example. We also have campaigns that work to phase out and ban harmful pesticides like chlorpyrifos, in which that was a successful campaign in 2019. We're quite happy about that. Glyphosate is still ongoing. Next slide, please. Our campaign also strives to raise awareness of the growing scientific evidence on how pesticides are harming our health. You see here on the right-hand side, a simple infographic that we developed on how harmful pesticides impact health and the environment, who is at risk, what exposure is associated to health harm, and what we need to phase it out. We create these types of visuals in multi-languages as we're across 27 different countries. And we do short videos to protect and to contribute to the health narrative and what we think increases both the frame and the drive for supporting a strong phase out of pesticides. We do webinars with scientists or medical doctors, or our members or experts, and then we share these with communities, our network, and our policymakers. Next slide. Here, I wanted just to share, underpin a bit about how we use science to raise awareness and our advocacy and ensure that everything we're doing is based on the latest science. So we work closely with researchers to share their findings on the health impacts of environmental risk factors. Here on the slide, you see the projects that we're currently involved in. Some of them are looking at the exposome, like IHEN, or others on EDCs and reproductive health, both male and female, like Merlon. Next slide. So now I just want to conclude, before I give over the floor to Rosella, I wanted to share a bit about what's the rationale behind why we chose to commission the report on men's health and chemical pollution. Since its inception, HIL has worked to really promote and protect the vulnerable from pollution and advocate policies to prevent exposure and to protect them, particularly at European level. We have done a lot of work on children, on pregnant women, and also on those most affected by pollution who already have diseases or are living socioeconomic deprived areas. In 2009, as you see here, so that was already 15 years ago, we did a brochure on men and on men's health under threat. And since then, we realized that there's been, really not a lot of looking at this issue. And we wanted to translate some of the new evidence on men's health and chemical pollution in Europe and look at what is the science telling us. So we also wanted to contribute to reaching out to new stakeholder groups focusing on men's health. One of them is Movember, which is a global campaign and that's why we launched it in November, that looks at prostate and testicular cancer, and also the Global Action on Men's Health. So I think we were very excited that Rosella was able to do this report, and she's going to tell you more about it in the future. And the next is just to show you that this report is available in several different languages, from the executive summary and the preface, and in particular Spanish, which you may be able to use. Sorry, we don't have Portuguese yet, but maybe one day. So you can find all of these on our website, and I really want to thank you for listening and happy to answer any questions either today or by email. Thanks.
Thank you so much, Anna. Next, we have Rosella Canarella, MD, PhD. Dr. Canarella is a certified clinical andrologist specializing in male reproductive and urinary systems, and an endocrinologist with extensive experience in male infertility and reproductive endocrinology. She earned her PhD in translational biomedicine from the University of Catania in Italy and expanded her training with fellowships at the Cleveland Clinic and the University of Massachusetts Medical School in the US, where she received her medical degree. She treats male infertility and endocrine disorders and conducts award-winning research. Dr. Canarella is chair of the Endocrine Society's Special Interest Group on Endocrine Disrupting Chemicals, member of its Special Program Committee, and serves on the Italian Ministry of Health's Technical Scientific Committee. She has authored over 260 peer-reviewed articles and is associate editor of the Journal of Assisted Reproduction and Genetics and other prestigious journals. She is the author of the recently released report, as Janon just said, by HEAL, called Chemical Pollution and Men's Health, a Hidden Crisis in Europe. Welcome, Rosella.
Good morning and good afternoon, everyone. So thank you, Mr. Feldman, for this nice presentation, and thanks to the Beyond Pesticide organization for inviting me to this lovely webinar. So first of all, I would like to say that I'm really excited to be here today. Because this is really a sensitive topic. It is something that needs to be spread to the community, scientific, etc. And because of the widespread men's crisis, I mean, you will see what I'm telling about afterwards. So next slide, please. I thank Janon and the HEAL organization for inviting me to write this report, which is focused basically on collecting the evidence that associate exposure to chemical and men's health. Next slide, please. So basically, I will not go through the report today because my goal here is to communicate and understand how basically endocrine disruptors can affect men's health. And of course, men's health means so many outcomes. So we will focus on specific outcomes just to, have a complete understanding of what's going on, basically. So you can anyways access the report and the explanation to it using this QR code. The report basically outlines the impact of chemicals on the epidemiology and the prevalence of testicular cancer and prostate cancer, male infertility, and even transgenerational health, and prioritize EU-based work, basically. It also outlines the issue related to the increasing prevalence of prostate cancer, testis cancer, and male infertility, and also their impact on the economical status, basically. Next slide. So what are we talking today is basically resumed in this agenda. So the report, the title of the report is a hidden, so it's Chemical Pollution and Men's Health, a Hidden Crisis in Europe. So first of all, I'd like you to, just to be on the same page, why we are talking of a crisis. Next slide. So I am a reproductive endocrinologist, and my research, as well as my clinical activity, is focused on men's health. And at one point of my career, basically, clinically and also on a research point of view, I was somehow forced to focus on endocrine disruptors because they are so widespread, and the epidemiology of infertility, of male infertility, was really connected with the exposure to chemicals. Next slide. So why crisis? You know that in 2023, the WHO has published an alert regarding couple infertility, mentioning that 1 in 6 people, basically, of reproductive age globally, so regardless of the country they come from, are affected from infertility. And I would like you to understand also what is the psychological burden that this couple may carry. Next slide. Okay, perfect. So in this P chart, basically, you can see a representation of the reason why infertile couples access in national ART centers for second and third level techniques. So first of all, if you look at all couples with infertility, we already know that a half of this couple suffer from infertility for a female factor infertility, a half from male factor infertility. So the prevalence is 50-50. So if we look at this P chart, this is assessing the prevalence, basically, of infertility in second and third level techniques. Next slide. And you can see that while the female factor of infertility are well and deeply characterized, so you can see how many female access ART centers for reduced ovarian reserve or for tubal factor infertility, etc. For male factor, you can see that there is just, a slice of this P, but there is no diagnostic characterization. And this is because this is something that is not common in Italy, because this data come from Italy, but these are really representative of a widespread all over the world. So in Europe and in the States, for example, as well. And the point is that the men's health, and specifically in this case, men's infertility or fertile status is totally under diagnosticated and under evaluated in the infertile couples. Next slide. This is really concerning because the evidence has already shown that globally, so regardless again of the country that we considered, the sperm count and concentration, so the capability of the man to produce spermatozoa, basically has declined, and particularly it has halved in years, which is really, really concerning. So we see clinically, and this data corroborate with the evidence that the prevalence of male factor infertility is really increasing. Next. And so the question that I want to answer, at least partially during this presentation, is what is in this scenario like? So in this declining, so what is the role that endocrine disruptors play in this? Please, next slide. So we will first need to understand what do we mean with the term endocrine disrupting chemical. Next slide. So according to current definitions, thank you. So I need you to consider three main take-home message when we talk of endocrine disruptors chemical. First of all, they are really, really widespread. So, for example, we can find them in cosmetics and personal care products. We can find them next in pesticides, as you all know, herbicides and fungicides. So these substances, for example, so pesticides contain bisphenol or parabens, and so they contain a mixture, basically, of endocrine disruptors. Next, we can find them in industrial chemicals, and this is the case for bisphenol, phthalate, and so on. And finally, we can find them in metals. So examples are lead, cadmium, mercury, or arsenic. But these are not the only sources. Food is another important important source. For example, in food, through food, we are exposed to phthalates, because phthalates are involved in the production of food, as well as in packaging on food. Same for bisphenol. They are involved both in production and in packaging. PFAS are involved in packaging, or nitrates, for example. They are important for as basically preservative for meat. Next, in the chapter of endocrine disruptors, there are also are worth to be mentioned, the so-called regrettable substances. So bisphenol S, for example, is an example, right? So bisphenol S has been introduced to replace bisphenol A. So it was first thought that it was safe, but now we know that it is not safe. It exposure to harmful effect like bisphenol A. So this is an example of a regrettable substances. And so far, many substances basically have been introduced to replace other, but many of them basically have been now included in this chapter. And next, something that is important is that we are exposed to a really wide range of chemicals through so many sources: the air, the skin, and the gastrointestinal tract, and so on. And we are not exposed to one substance at a time. And not to 100 of substances at a time. We are talking of thousands of chemicals to which we are simultaneously exposed. Next. So this allows me to introduce the second message. That is the problem of the mixtures. So the fact that we are simultaneously exposed to so many substances create basically a synergic or an additive effect. So to understand how this works, for example, one substance is able to increase, for example, the receptor to a second chemical, and therefore the body, because of the exposure to the first chemical, become sensitive also to the second one. And therefore, this may create a kind of additive effect. Next slide. So the third issue regards the effect. So basically, endocrine disruptors classically are known to interfere with hormones, basically, and so with endocrine glands, and the testis is one of them. And therefore, the classification introduced by WHO that has been used by policymakers as well, basically classify the endocrine disruptors as, for their capability to disrupt hormone balance. But the problem is that this is not only, this is not the only mechanism through which they are harmful, and therefore this can create issues when we have to identify a specific and target a specific chemical that is an ADC, but biologically is not, or for policymakers, for example, is not considered an ADC because it does not disrupt the hormone balance. So some chemicals, for example, do not have a great effect on hormone balance, but they are able to influence the epigenetic of a cell. And this is important for the issue of transgenerational inheritance when this happens in the spermatozoa, because this epigenetic modification is transmitted and inherited by the embryo. And also they are able to influence the fate, basically, of a cell, for example, inhibiting signals of apoptosis, and therefore, resulting in an increase in cell proliferation. And this is the reason why this may, promote cancerogenesis. Next. So we have basically, next again, thanks. So we basically have introduced the most important aspects regarding endocrine disrupting chemicals. Now we want to know what's, their role on spermatogenesis. Next slide, please. So when we introduce this topic, first of all, we need to understand that timing matters, because the exposure during prenatal life has a different effect compared to exposure in adult life. And this is because the testis is made of cells that are still immature, are still differentiating during prenatal life and during early childhood. And therefore, when there is an exposure at that age, the impact basically in adulthood will be an infertility and a reduction in sperm production that unfortunately is not reversible. Okay, when the exposure happen during the adulthood is a different story, because spermatogenesis is capable of replacing, of renewing basically every 2 to 3 months, and therefore quitting basically from the exposure is able to repristinate the normal spermatogenesis. Next. So the way through which chemicals are able to influence spermatogenesis is, first, endocrine disruptors are capable of influence hormones, and therefore the hormonal regulation of spermatogenesis. This is not the only mechanism, because chemicals are able also to cause inflammation in the testis, and the sperm are cells that are really sensitive to oxidative stress, inflammation, and this causes damage in the DNA of the sperm that in turn led to infertility. Third, chemical may also, as we said earlier, alter the epigenetics of the sperm, and this is important because it has transgenerational effect. Next. So this is a very important study. This is a epidemiologic study, basically, and a longitudinal one. This is a kind of example, so a study that I, present really to catch and understand the impact of these chemicals on infertility, on male infertility. And particularly in that study, the investigators have evaluated maternal exposure to PFAS during early pregnancy. And then they evaluated basically their sons during reproductive age, so at 20 years old, and they evaluated where the sperm production was impacted and was related somehow to the levels of PFAS to which the mother were exposed during the first trimester. Next, something that was very interesting was the confirmation of the presence of not only of an effect, but also of a dose-dependent effect, and particularly for a 1 unit increase in exposure to PFAS, that PFAS include a range of chemicals, there was a reduction by 8% of the sperm concentration and of the total sperm count that is not reversible anymore, since the exposure happened during pregnancy, basically. And this was seen in the young man. Now, next, I want to understand whether this is just an association of if there is a causation. Next. So this animal study is really interesting because basically mice during the early life basically have been exposed to a mixture of chemicals that somehow was designed to simulate the mixture to which we are exposed. And the authors basically observed that the exposure during the childhood life in these mice led to infertility in the adult age, and this was mediated by an alteration of the metabolism of the Sertoli cells that are capable to support basically the spermatogenesis. Next slide, please. And therefore, this study basically demonstrates a causal effect, because the Sertoli cells still in childhood, in human life, basically, basically are immature and capable of proliferating until puberty, while they lose the capability of proliferating when puberty begins, and they acquire the capability of stimulating germ cells. Now, each Sertoli cell is capable of stimulating a specific number of germ cells. So if the child, basically the kid, reach puberty with a reduced pool of Sertoli cells, this reduced pool will be able to stimulate in turn a reduced pool of germ cells, leading to a non-reversible lower sperm count. Not reversible because Sertoli cells cannot proliferate anymore. Therefore, if endocrine disruptors during fetal life and early childhood are able, and this is a really key point, are able to influence to reduce the Sertoli cell proliferation, this will lead in the adult life basically to a non-reversible reduction in sperm production. And this has been observed and demonstrated in animals. And therefore, we can conclude that it is not an association. It is a causation link. Next slide, please. Next again. So now that we got basically this aspect and so have assessed the link between EDC and sperm production, I want to briefly introduce the issue related to transgenerational health. Next. So coming to the assessment of this link, so far, what we are exposed to, like the information that we already know, and it is, of course, evidence-based, is that maternal exposure led to a series of consequences in their kids. And in the male newborn, this will affect basically the testicular function and reproductive life. So basically, all transgenerational health is focused on the maternal exposure. But what about paternal exposure? Next slide, please. This is a really important question because so far, the sperm has been considered just a carrier of paternal DNA. But recent evidence, and I'm referring to evidence that has been published in the last 15 years, has revealed that the sperm is not just a carrier of paternal DNA, it carries also RNA, and also the DNA has specific imprints that all are transferred to the offspring and are capable of influencing the health of the offspring, predisposing the offspring itself to developing specific disease. And next slide, please. And I want to share with you a really simple, example to get this important aspect. For example, in this study, two groups of mice were exposed to controlled diet and to a high-fat diet. Then they were, after mating, basically the pups at 6-24 weeks were sacrificed to see basically, their metabolic health. So they wanted to know whether the exposure to the high-fat diet in the father was able to influence the metabolic health of the offspring. Next, what the author found basically was that the weight of the pups of the two groups was not affected, so they were comparable, there were not obesity, but there was an increase in glucose level of the pups in the group of the pups that were conceived from the mice exposed to the high-fat diet. Next slide, please. And the same pups also had changes basically in the liver metabolism. Next again. They basically found that there was an increased expression in an enzyme that is called the phosphenol pyruvate carboxygenase that trigger gluconeogenesis and therefore increased glucose levels in the blood. This increased expression was due to an increased expression of a gene that is called H19. Next slide. And the authors basically next demonstrated that this increased H19 expression in the liver was the cause of the increased expression of the phosphenol pyruvate carboxygenase in the liver. Next slide. The interesting point comes when they basically try to understand why the H19 gene was hyperexpressed. And what they found is that the hyperexpression came from an abnormal methylation of the H19 in the liver of the pups. And this abnormal methylation came from an abnormal methylation of the sperm. So what happened next was that the father exposed to a high-fat diet basically also carried dysregulation in the methylation of the H19, which is imprinted. These abnormalities in the epigenetic was transmitted to the offspring, and therefore the offspring developed higher glucose levels. And this was carried by the abnormality in the sperm. Next. In human, it was recently demonstrated that basically a greater paternal BMI is associated with an increased levels of glucose and insulin, which in the kids of 5 years old, which absolutely mirrors what the evidence in animals has found. Next. So this aspect, this evidence that is coming regarding the epigenetic role of the transgenerational inheritance coming from the father, from the epigenetics abnormalities in the epigenetic of the sperm can be translated also with the exposure, the paternal exposure to the endocrine disruptors. Next. A very interesting review has been published on that, where a total of 19 papers have been reviewed, and they all carry evidence that is really concerning. This is extended to mammals and not mammals, so this worryingly may regard the human being as well. And what the authors basically conclude is that the exposure to a wide range of chemicals, basically in the paternal life, particularly in preconceptional window, is associated to the susceptibility of the offspring to develop reproductive toxicity or neurotoxicity or skeletal deformity or metabolic disorders. So this is concerning because if we translate this in the human, in the paternal, if we consider preconceptional paternal exposure to endocrine disruptors, this exposure may affect not just the health of the father itself, but also the health of the sons or of the daughters that may be exposed to a higher increase of developing specific disease. Next. For example, in this study, which is again on animals, paternal exposure to cadmium and high-fat diet caused an abnormality in the sperm epigenetics that basically was transmitted to the offspring and that was associated with neurodevelopmental disorders. So if we consider the increase in the prevalence of autism, this data are concerning because there may be an association between this epidemiologic aspect and the exposure to chemicals. Next, for time reasons, we will not, next again, thank you, we will not talk about testicular cancer or prostate cancer. So what we can do, this may be, subject of our discussion, but I wanna say that there are interventions that are individual, so that depends on can be introduced in the daily life of every person, basically, and that can minimize the exposure. Of course, there are other interventions that depends on the institution, and there are several limitations so far that prevent a comprehensive evaluation of endocrine disruptors. For example, there should be more sensitivity to detect what are the sensitive windows of exposure, and preconceptional life for father is one of those. Second, the issue of mixtures. So the point is that currently we measure one chemical at a time, but we should measure the mixture of chemical. Third, the problem of identification. Not all EDC are correctly identified. And this is another issue because they are considered EDC only those that determine hormonal impact, hormonal alterations, but these are not the only chemicals to which we are exposed to, and therefore this can create issues in the proper identification of these chemicals. So thank you next again. Thank you so much for listening, and I'm open to any question, and I'm open to reply now or via email as well. Thank you so much.
Wow. Thank you so much for the presentation, and both Carolina and Rosella. Your deep research is so appreciated, and you're cutting important ground here that we all hope we can bring to the policy context, and that we'll see some effect. I know we will. This is incredibly important research. So with that, thank you again, Rosella. We're going to move through the program now. Carolina Panis is a PhD, is an associate professor of medicine at Western Paraná State University in Brazil, and visiting researcher at Harvard and the University of Arizona. She holds postdoctoral fellowships in oncology and pathology, focusing on breast cancer, genomics, immunopathology, and chemoresistance. Her research investigates the impact of pesticide exposure on cancer aggressiveness and tumor immunopathology mechanisms. Dr. Panis coordinates the tumor biology laboratory and the micropollutant residue analysis laboratory at her university, and collaborates with leading institutions in Brazil and abroad. Dr. Panis runs a laboratory that researches cancer biology, focuses on human breast cancer and hematological neoplasia. Most of the studies have investigated the role of oxidative stress and inflammation and the environmental risk factors that may determine disease prognosis, especially in rural women occupationally exposed to pesticides. She published Pesticide Exposure and Increased Breast Cancer Risk in Women Population Studies last year. Dr. Panis received her PhD in experimental pathology from Londrina State University. Welcome, Carolina.
Good afternoon. I would like to thank you for the opportunity of being part of this amazing event with such an important group of speakers from different regions of the world. I would also like to thank the interpreters and the whole team that supported Jay in this process. You were all very kind in bringing us to speak here today. So as Jay has said, I'm going to talk to you a little bit about the Brazilian experience we have in terms of characterizing the intoxication by pesticides that is happening in women who are exposed, occupationally exposed. So my focus is breast cancer, but we have been developing studies with different populations, such as the children population, men with intestine cancer, also lung cancer. So we have a long list of characterization during the last decade. So the data that I'm going to bring to you today, I'm going to talk about breast cancer only. And why are we going to talk about breast cancer specifically? Because it's very relevant. It's the most common malignant neoplasia in women. We know that it's a very known fact that 1 out of 8 women will develop breast cancer during their lives. And there are many risk factors, and many of them are connected to the endocrine disruptors. So this is well known that anywhere in the world, age, genetic factors, family history, breast density, overweight, sedentarianism, alcohol intake are important factors for the development of this disease. However, what we've realized over the years is that even with these well-known factors, many of them such as hormonal medication, smoking, connected to some endocrine disruptions, still, we cannot explain the increase of the number of breast cancer cases over time. There is an improvement of healthcare centers to do this diagnosis. Access to healthcare has improved, but still, the number of cases has increased a lot. So, one thing that caught our attention was that these classic risk factors do not explain the high rate found in some populations, such as what we see in Brazil, especially in my state. So, Brazil is a very large country with a very large territory, and the southern region of the country is a region of high incidence of breast cancer. And that's where I conducted my study. So we started to think that there could be additional factors to explain the number of breast cancer cases. So 1 decade ago, we started a study. If you're interested, you can take a look at it. It is summarized in this Lancet publication. We were the editorial in the January issue. So our study was highlighted as an important study in breast cancer, especially because it brings this connection between the increased risk of developing breast cancer and pesticides. So just to detail to you what we've been doing here and why we are studying women farmers. So on this map, this is the map of Brazil. This is a map that shows the volume of consumption of pesticides. We have the average of the last few years. So the darker the color, the higher the consumption. Brazil has a consumption average of 6 kilograms per capita.
And here you can see that in my state, Paraná, which is represented by letters P and R, the third state from the bottom up here on the map, is a state that consumes a lot of pesticides because of the production of food. Paraná is the top food producing state in the country. Everything that is exported to the US and Europe is food produced in the state of Paraná, and therefore family farming is very important in this region. So here in Brazil, you can see that we are the second largest agricultural production value in the state. And our agriculture here is characterized by family farming. People who know that Brazil is really strong in agriculture think that Brazil only has mechanized agriculture or agriculture like we have in the Midwest of the country with huge machinery and pesticides being sprayed through machinery. But actually, in the region where we studied, people spray pesticides with small pumps, just little boxes that are put on the back of farmers with a little device for spraying those pesticides. So they work for 8 to 10 hours a day just spraying pesticides on the fields. And I want to bring your attention here to the pesticide mixtures. In Brazil, just like in China and the US, because we are top pesticide consuming countries, we have a predominance of 2,4-D and other endocrine disruptors. So we wonder, how do women get contaminated? And with Professor Bernardo Lemos from University of Arizona, we conducted a survey to try and understand how women participate in these processes. And the literature does not offer us any clarity. So we decided to focus our research on women exposed to pesticides, because if you go to PubMed to search for breast cancer, pesticide, women, and other similar keywords, you see that there are about 600 papers that supposedly focused on this topic. But when you actually filter, you realize that only 11 studies actually assessed women's exposure. And of these trials, none gives us any information about the disease profile of those women. That's why we found it would be important to focus on this research topic. Now, take a look at the breakdown of the state of Paraná. The population that we study lives in 27 different municipalities that you can see on the map, in which the pesticide consumption is up to 70 kilograms per capita, 10 times more than the consumption of the state of Paraná overall. When we started the trial a decade ago, we saw that the breast cancer incidence was really high in this region. Over 40% of the incidence that we see throughout the country. I mean, over 40% the national average. And the mortality rate is also higher, about 14% higher than the rest of Brazil. And breast cancer can be cured. In up 95 to 99% of the cases that are identified early, this can be cured. So in operable breast cancer, I mean, these women here in this case have a very high mortality. So we see high pesticide consumption, high incidence, and high mortality in this population. So we thought, is there a correlation among these factors? That's when we started to characterize the exposure. We call this occupational or domestic exposure. This is actually a chronic, continuous exposure that happens on a daily basis. These women handle and manipulate pesticides every day. And throughout the process, we interviewed almost 600 women, and we saw that the main exposure route of these women in the municipalities that we studied were laundry. Laundry happened in 94% of the cases that we interviewed, and they were washing their clothes without any type of protection gloves. So these women were being exposed to the chemicals without any type of protection. So they do get contaminated throughout this process. So then I went to the Harvard University in the public health school, working with Professor Bernardo Lemos and Dr. Marcia Castro. And we decided to run a study with the data that I had collected from Brazil. So we looked at the cohort of patients that we had, and we ran a study of women that were exposed or were not exposed with and without breast cancer. So we got samples from these women to assess contamination, and we also evaluated the clinical profile of the disease. This is a map of the region that we studied. The yellow dot in the map indicates where these women live. So they are completely inserted in farming land. Their houses are also sprayed, so their houses are highly contaminated with pesticides as well. And we invited these women to get their urine samples, and we identified that in women who lived in areas 3, 9, and 6, we saw not only contamination, that you can see here in the first column with glyphosate and atrazine, but also in property 6, for example, it's only the women who are contaminated with glyphosate. And the sprayer, who's usually the husband, and that you can see here in the second column, in this case of property 6, he was not contaminated because he used individual protection equipment. So we see that the skin absorbs the pesticides. All of these women got their samples taken after they did the laundry. So we saw that while they were doing the laundry, the pesticides would go into their skin. This is a picture of our lab. Okay, this isn't from any publication, but we compared the urine of control women from urban areas to compare to the urine of exposed women. And we can see that the urine from exposed women look completely different. And when we take the urine for a metabolite dosing of glyphosate, we see that there is a very high concentration after they do the laundry that can get up to 1,000 times more the concentration that we see in women from urban areas. So for us, it's very clear that laundry is a source of contamination to these women. And in this population, we checked the risk of developing breast cancer comparing exposed and non-exposed women, and we saw a higher risk, actually 52% higher risk of developing breast cancer and also metastasis, which got to 219% more in women that are exposed to pesticides and are contaminated with glyphosate. And when we talk about breast cancer, we have to think about two things. If there is a carcinogen or an endocrine disruptor or a mixture of components that are leading to the disease. But once the breast cancer has developed, we have to think about how it's going to be eliminated from our body. So this is a famous hallmark of cancer. These hallmarks explain the characteristics of cancer, but the most important hallmark would be avoiding immune destruction. So we decided to look into the data to see if the destruction was happening in contaminated women. I'll just summarize the findings. I'm not going to go into details about the molecules, but we have a huge set of soluble components and cells that participate in the process of eliminating the cancer cells. And we looked at the key components to eliminate breast cancer. So during the pandemic, we developed our first trial tracking blood samples from these women and comparing exposed and non-exposed women. And in exposed women, we saw that there was a downregulation of their immune system. Their immune system and their components are highly reduced when they are exposed to pesticides, especially the alpha tumor necrosis factor or TNF alpha, which is reduced in the blood of women exposed to pesticides. So, because of that, we decided to look into other molecules. Here I have an example of interleukin 12, which is a very important molecule for anti-tumor immune response, and that is always depleted in these women. So now I have a tissue database and clinical database of 1,800 women, and all of the samples of farmers, without exception, do not have interleukin 12, which suggests that the absence of this substance can be leading to this down regulation because of the exposure to glyphosate, atrazine, and 2,4-D. So here you can see breast tumors, first set for non-exposed women and second exposed women. And here you can see molecules TGF-beta and CTLA4. And we can see that exposed women have a higher expression of these molecules compared to women who were not exposed. Now, all of these proteins I mentioned, like TNF and interleukin 12, are important regulators of immune infiltrates. If the immune system doesn't go into the tumor, there is no elimination of cancerous cells. And we see that there is almost no lymphocyte infiltrate of TCD8 and a disorderly infiltrate of TCD4 in patients exposed to pesticides.
Suggesting a scenario that is known as an immunologic desert. The worst tumors, they have this desert, this absence of immune cells that will be responsible for the recurrence, the treatment failure, and problems with the patient long term. So these are images of what immunologic desert is. This is a tumor of an exposed woman, and the right side is an exposed woman, and the left side is unexposed. So this, if good prognosis, is linked to leukocytes. So we were thinking if there was no tissue immune exhaustion in these women, and we proved that there is indeed. We have data of not only interleukin 12 and TNF alpha, but other factors are also impaired, and also in tumor tissue. So there are immunologic problems. There are no production of tumor killing chemicals. So we found that women who do not produce these substances, these chemicals, that they are young. They are diagnosed before 50 years old. And if we check the two charts, we can see that they have completely opposite. If you look at the left-hand side chart showing diagnosis above 50 years old, which is the expected for breast cancer, compared to the right-hand side one that shows the young women, we see that there is cytokines profile that is similar to pesticide exposure that's completely different from one group to the other, showing that the depletion of interleukin 12 is highly associated to exposure. In the same group, we showed the occurrence of a different profile, different from the rest of the world. And breast cancer in general has good prognosis, and we call it luminal A. And here in our population exposed to pesticides, we find that women are premenopausal young women, and they have luminal B tumors, which are metastatic and highly with high proliferation. So we've seen that there are some findings connected to endocrine disruption. And we find that women who are exposed to pesticides, they have a disruption of cortisol, for example, and also insulin. This has not yet been published, but we can see that there is an important disruption, a significant disruption. Other hormones are being studied, but women who are exposed to pesticides have a high toxicity, immunoendocrine toxicity profile. And this is the sad part. The saddest part is that if we look at this shows us how mortality in women develops all over the world with breast cancer, and we find that this mortality has been decreasing. But when we think about the population exposed to pesticides, and we conducted a study to show this, we don't see this mortality reduction trend. In this study, we studied women with operable breast cancer with cure rate between 95 to 99%, and we found that the cure rate in farmers is below 80%, even in very curable cancers. And because of the exposure to pesticides, we find that these women end up having a bad prognosis. So they do not respond well to surgical treatment. And to conclude, I would like to say that we've been conducting many studies connected to exposure, not only occupational exposure, but also environmental exposure. And I think that because of time constraints, if you're interested, we can talk about this further during the Q&A session. But we've shown a high a correlation between this exposure and the increase in cancer cases in this region in Brazil. So we've done many studies with these women to educate, teach these women that they cannot wash their clothes unprotected, without protection. They cannot expose themselves to pesticides. And we've been talking to the government as well. We've been trying to regulate protection policy for women, because women are an invisible element in this pesticide chain. We don't have policies directed towards women's health. And in the last administrations were denialists and did not look at the pesticide issue. And we know everybody knows that Brazil is one of the highest users of pesticides. Pesticides that are prohibited all over the world are still used in Brazil. So we have this void, we have a challenge, and this is a political challenge. We need to sensitize our managers, regulatory agencies for this theme, because they need to see women as part of this process. This is what I wanted to say. Thank you. I would like to thank you for your attention, and I'm available for the Q&A session. Thank you very much.
Thank you, Carolina. Extraordinary work. We're going to move through the program now on to Jabin Tiba, PhD, who is a postdoctoral research associate in Water, Climate, and Health program at the University of Nebraska Medical Center. Earlier this year, Dr. Tiba published a paper called Exploring the Joint Association Between Agrochemical Mixtures and Pediatric Cancer. Dr. Tiba is formally trained as a multidisciplinary researcher and epidemiologist and has prior experience in clinical research, public health practice settings, and using large data sets. Her research interests include understanding the impact of environmental exposures on the genome to protect human health. Her current research is primarily focused on understanding the effects of environmental mixtures, which we've heard a lot about today, on human health, specifically exploring relationships between pesticide mixtures and pediatric cancer, congenital abnormalities, and other health outcomes. Dr. Tiba received her PhD from the Medical Sciences Interdepartmental Area Program at the University of Nebraska Medical Center after receiving an MPH from the University of Cincinnati College of Medicine. Welcome, Jabin. Really appreciate you being here.
Thank you, everyone, and thank you, Jay, for inviting me to this forum. So I'm very excited to share with you all about the research that I've been doing. So today I'm here to talk primarily about my research that is focused on agrochemical mixtures and pediatric cancer. Just a little bit about the agenda. I would be talking about the introduction and giving you a little bit background about what motivated me to do this work, and we'll be discussing the methods that I used and our results. So talking about the pesticides, like we've been hearing since this forum started about different kind of environmental chemicals. So these pesticides, these are the endocrine disrupting chemicals actually. So these have been like widely used. So I want to emphasize about the application of these pesticides globally. So this is the most recent Food and Agricultural Organization statistics. So based on this statistics, from in the past few decades, the pesticide use has been like very high. So if you look on the countries that are highlighted in red, So these are the countries where the pesticides are very highly applied. So unfortunately, even United States happens to be that number one country. If you look at the top 10 countries where pesticides have been used highly for agriculture, United States happens to be the number one country based on these statistics, actually. And so as we are talking about today, this talk is mainly focused on Nebraska, but even before we go into Nebraska, I want to just show, like, how the agricultural land use had been in this country. So since like 1990 to 2014, I am sticking to this timeline because this is the timeline of my study. So I just want to point out to the agricultural land use in this country. This is a picture that is created using Google Earth Engine. So the areas in the green, mostly in the central part of this country and some parts of the California and the western states of the United States, like there is a lot of crop land that has been used for agriculture. So this also happens to correlate with the pesticide application. In our previous study, we were looking at the risk, we were identifying the risk-based areas for pesticide application based on the social vulnerability. And this map of agrochemical land use happens to be correlating with the pesticide application. So more focusing on the Nebraska because our research is in the Nebraska. So Nebraska is the heartland of this country where a lot of like corn and soybean is produced. So the counties in the Nebraska, which are in the darker green and yellowish regions, these are the counties where there is like a lot of agriculture happening. So the topmost portion, like the northernmost part of the Nebraska, like we don't have a lot of agriculture there, but these are the counties where there is like a lot of agriculture is happening. So when there is a lot of crop production, pesticides have also been consistently used in all these Nebraskan counties, actually. So yes, why are we concerned about pesticides? So what if they use the pesticides on the croplands? Pesticides might be increasing your produce, but pesticide is also contributing to a lot of environmental health burden. I want to emphasize on this picture, this was taken from Rachel Carlson's book on Silent Spring. She wrote this book in 1962. So her work on pesticides, its toxic effects in the environment, bioaccumulation of these pesticides, and not only impacting the wildlife and humans, this book, like this revolutionized the environmental movement in this nation, actually. So after her work, DDT was banned in this country. And then following her work on Silent Spring in 1962, this not only led to the modern environmental movement, but also in 1970, that's when the EPA was also established. So this book, actually, her work contributed to that. I just want to reiterate that. And yes, so yes, pesticides are bioaccumulated. They are in our atmosphere. They are in the places where we live. But how are we exposed to these pesticides? So we are mainly exposed to these pesticides through inhalation, through the air we breathe, through the ingestion, like through drinking water and the food, and also through dermal contact. So in the previous picture, I was talking about the bioaccumulation of these pesticides. So yes, once these pesticides are applied on the cropland, they're not only bioaccumulating, polluting the soil, but also agricultural drift happens. So that's how through the air, these pesticides are spreading. So that's how we are like inhaling those pesticides. And pesticides, these are also when these cattle which graze on these croplands, so the pesticides are also entering, and they are bioaccumulating in different organs and organ systems of the cattle. And these pesticides, they further drift and percolate also into the groundwater systems, polluting our aquifers and the water. water bodies. So we are, like general public, we are exposed to pesticides through the food we eat, through the water we drink, and that just doesn't stop there, because but there are also farmland workers and people who work on the crop plants, like, they are also constantly exposing to the pesticides uh, due to if they might be wearing the personal protective equipment or not. Based on that, they are being exposed, and those pesticides, they are still on their clothing. So why we are also talking about that, because my topic is mainly focused on pediatric cancer. So which is why I also want to talk about the occupational exposures, because children, they are so unique and they are so vulnerable. They're not only just exposed to the food, but also through the parental exposure is also very important. So which is why I want to emphasize even on the occupational exposure. So exposure to pesticides happens even before birth. So some of my colleagues here, they were talking about prenatal exposures. So yes, it could happen during the pregnancy or even before conception. And even children are also exposed to these pesticides in their different environments they live in. Their built-in environments also matter. So that's how the commonly children and in general, we humans, we are exposed to these pesticides. So what we are exposed to these pesticides, my colleague Rosella, she was talking about different, characteristics of the endocrine disrupting chemicals. So because of these endocrine disrupting nature of these pesticides, so these pesticides are not only causing cancer, but they are also like mutagenic. They are toxicating our genome. They are causing different health effects, not only limited to the cancers, but also like neurotoxic, neurodegeneration, neurodevelopmental disorders. So these are some of the pathways of how these pesticides are exposed and the kind of toxic health effects they're causing in our children. So this is just, again, a little bit background on the mechanism of how this cancer develops. So someone is exposed to a pesticide. So one of the characteristic of this endocrine disrupting chemical is like it's causing epigenetic alterations. So it's causing DNA damage, or like damaging the histone. So when this DNA damage occurs, like, when that is affecting the cell, it further proliferates, and it could be either leading to apoptosis, or it could like further that the damaged cell, when it further progresses, like, due to the genomic instability and due to the loss of tumor suppressor genes, so it's causing cancer. So this is just the basic mechanism on how this pesticide exposure is causing cancer. So the motivation for our study came from an environmental contamination that happened in the place where I live in. So we had an Alton plant, which is a bioenergy processing plant. So that there was a contamination due to neonicotinoids. Neonicotinoids are like a replacement of organophosphate pesticides. So my interest began in this area since that time. And also, as we were talking a lot about mixtures, so as I happened to learn more about the pesticides, I also learned that we are not just exposed to one chemical, but we are exposed to a mixture of chemicals. And NIH's emphasis and focus on these chemical mixtures also, and the development of like epidemiological methods in this area of mixtures also motivated me to pursue the mixtures actually. And additionally, cancer is the top leading cause. It's the second most leading cause of death among children, especially the children who are aged between 1 to 14 years. They commonly, childhood cancers is the leading cause of death among them. This is in the country, but in Nebraska, the cancer incidence and cancer mortality due to pediatric cancer among children is slightly higher than the national scale. So this high burden of pediatric cancer in this state, it actually motivated me to study about pediatric cancer. So previously, previous research was done in Nebraska, but this was like mostly limited to single chemicals like atrazine or nitrate. So this led me to look at and consider integrating like more chemicals to study their co-exposures within the chemical mixture and to estimate the overall estimate or overall burden of those agri-chemicals on the pediatric cancer rate. So the main objective of our research was to identify the high-risk Nebraskan counties, firstly for high pesticide exposure and high pediatric cancer rate. And then we wanted to estimate the overall joint effect, like the cumulative effect of all these pesticides after adjusting for their social vulnerability factors. Because social vulnerability was important because, like, social vulnerability not only influences or impacts the pesticide exposure, but also the cancer vulnerability as well. So, which is why we adjusted for certain social vulnerable factors, and also we just not only were interested to look at the overall effect estimate, but we also wanted to identify what were the top players, like, what were the top chemicals that were linked to this pesticide exposure, linked to this pediatric cancer. So ours is a cross-sectional study. So we are not talking about any causation, but we are only talking about associations. So we used pesticide application data from the USGS Pesticide National Synthesis Project. So that releases like their annual estimates for pesticide application on cropland per acreage. And this is like a county scale data. And our pediatric cancer data was obtained from Nebraska Cancer Registry. And for social vulnerability, we consider taking the US Census Bureau, like ACS Community Survey. 5-year estimates were taken. And yes, our spatial data was retrieved using a Tidy Census R package. And this was for the year 2010 because that was like the closest year. So we took that as we took that year for our spatial data. So the pediatric cancer cases that were included in the study were between 0 to 19 years, and these were based upon their ICD-10 diagnosis codes. And we have also used pediatric population per county as an offset variable while we were estimating the prevalence rate for pediatric cancer, as also for our overall effect in our joint models, in our mixture models. That was used as an offset variable, which is nothing but the denominator. So the social vulnerability factors that we considered were like race of the children. So our racial group, ethnic group, mainly comprised of non-Hispanic, Black, and like Hispanic or Latino. We adjusted for several of the economic factors, education and healthcare access. So these were the main factors that we considered for our social vulnerability. So, excuse me. So talking about the pesticide mixture, we included like 32 agri-chemicals. Like these were the top pesticides that were applied across the 93 Nebraska counties. So how we selected these top chemicals were we developed a specific selection criteria. Like we thought that, hey, these pesticides had to be applied in at least like 90% of the during the 90% of the time period of our study time period. And then this should be applied consistently in all the Nebraskan counties. So based upon this criteria, we have identified the top chemicals that were applied in the Nebraska croplands. And then we identified the high-risk counties for pesticide exposure and pediatric cancer rate by using the specific IPCC category was used to map and identify the high-risk counties. And we also did a principal component analysis and we developed a pesticide index to map the high-risk counties. And our mixture analysis was done using generalized weighted quantile sum regression model. So this is one of the mixtures model. And we have used a repeated holdout validation approach to estimate the overall mixture effect of this. So I just want to go a little bit about this model. So this is the most commonly used model. So one of the advantage of this model is that this supports our count data because our pediatric cancer cases were like counts. So this model supports like Poisson and negative binomial models, and it also supports zero inflation models as well. And it supports, because all these pesticides, these are like very highly correlated. So this model allows for using when you have a very highly correlated. And another advantage of this model is it not only gives you the overall mixture effect, but it also helps you to identify which chemicals were actually causing that effect, which were the top chemicals that would be contributing to those joint associations. So here is how much these chemicals were correlated. So I told you that we identified like 32 top chemicals. So of these 32 top chemicals, predominantly were like herbicides, of which these are like 2,4-D, acetochlor, alachlor, atrazine. These are like some of the top chemicals actually, and also followed by insecticides. So terbufos, tefluthrin, permethrin, epsom valerate. These were some of the top among all these 32 top chemicals that were used as a mixture for our study. As you see, all these chemicals, almost all these are like very highly correlated within the class of their herbicides and also within the class of the insecticides. So we have included like all these chemicals in our mixture. So these are the results from our spatial distribution of the top applied pesticides. So as I told you, our first objective was to identify the pesticides that were identify the high-risk counties in Nebraska that were at a high risk for pesticides, actually, where pesticides were like predominantly applied. So I want you to look onto the counties that are in the yellow color. So these yellow color counties are the counties where like the pesticides were highly applied. If you remember the previous map initially when I showed you the agrichemical land use, that actually correlates with this high pesticide application, because the counties in the purple, these are like very low chemical application, pesticide application, because these counties where there was not of like cropland was used. So this just correlates with my previous agrichemical land use map that I showed you. So we did this principal component analysis just to first identify that and develop a pesticide index, because we wanted to use this pesticide index to identify the counties in Nebraska that are at a high risk for not only just the pesticide application, but also the pediatric cancer rate. So in our cancer data, like the most common type of cancers were like the CNS cancer, leukemia, lymphoma, and some of the bone tumors. These happen to be the top cancer types within our study. And also in Nebraska, these are the top cancers, actually. So we wanted to identify the high-risk counties that are at a high risk for these different predominant cancer types in the Nebraska. So I want you to, when you look at this first figure, so this is like a bivariate map showing the pesticide application as well as like pediatric cancer rate. The counties that are in the darkest brown are these counties which had like high pesticide application as well as like high overall pediatric cancer rate. So Holt County, Platte County, these are like the Box County, Boone County, Hamilton and Butler counties. Like these are some of the high risk counties for this overall pediatric cancer. But when we look into the central nervous system tumors, because central nervous system tumors, these are not only the common or common prevalent type in the country, but also these happen to be the prevalent type in the state of Nebraska as well. So even here, some of these counties, the counties of Holt, Platte, Butte counties, Knox and Butler, like some of these counties, they still coincide with the high central nervous system tumor cancer incidence rate. And for coming to the leukemia, so even for leukemia, we found that like some of the counties, they still were consistent across these different cancer types, but we found that the counties, like some of these counties, like they are very high agricultural counties as well. So Lincoln County, Dodge County, Boone County. So these are like some of the newer counties besides the one that I discussed previously, the Holt, Pierce, and Platte counties. So these had like higher risk for pesticide application and higher risk for leukemia. And my last figure, this is on the lymphoma. So the counties here that are in the darkest brown, the Cass County, Platte, Bucks County, Madison, Lincoln, and Hamilton and Perkins counties. So these counties are at a high risk for high pesticide application, as well as like for high lymphoma rate. So coming to the main results of our joint associations between the agrochemical mixtures and pediatric cancer. So we have used like 100 bootstrap samples. This is like a method that is a part of the GWQS model, a mixture model that we used. And we used like 5 repeated holdout validations. And we have included 32 agrochemicals. And we have included all the cancer types that were in our study. So as I told you that on this x-axis, when you look at the x-axis, you see like total cancer, brain and CNS cancer, leukemia, lymphoma, malignant bone and germ cell tumors. So these tumors, they showed like some of these showed like significant associations, and some of these, like the malignant and germ cell, they did not show statistically significant association, but I would be explaining it more detail. So we constrained the model, as well as we used an unconstrained setting in the model. So constraining is nothing but you're just telling the model to run in the specific direction of your hypothesis. So our hypothesis was that when someone is exposed to cancer, there is increase in the pediatric cancer rate. So that is the positive direction. So when we constrain the model and when we unconstrain the model, so the blue color, the results that are shown in the blue, these are the significant associations, and those shown in the orange are the non-significant associations. So for those that we constrain the model, you see there is like a dot in the center of this line, and the triangle, the triangle that you see here on this line is like unconstrained results. On the y-axis, you see the beta estimates and the 95% confidence intervals, because the GWQS model gives us the overall effect estimate, and it gives us the 95% confidence interval. So our total cancer, brain and CNS type, and leukemia, they showed like significant positive association between the agrochemical mixture and the specific pediatric cancer rate, whereas the malignant bone tumor and germ cell cancers, they showed like negative associations. So the results were not like significant. So our key findings are like...
We're going to have to wrap up in a minute. That's possible.
Sure, yes.
Thank
So
you.
our key findings are like the herbicides like 2,4-D, atrazine, alachlor, and glyphosate, these were like most frequently applied. And we found the brain and CNS tumors, leukemia, lymphoma, and germ cell tumors. These were the top tumors. And as I already told you, the cancers that were like, that showed the positive associations. And we found that the independent chemicals, dicamba, glyphosate, paraquat, quizalofop, and trisulfiron and teflutrin, these contributed to the significant weight for our mixtures, actually. So yeah, since it's a cross-sectional study, that is one of the limitation. We could only talk about the association and not the causation, and we could not evaluate the toxicological aspects of our chemicals. And due to the limited sample size, some of the cancer types could not converge. So our future directions are to establish a human biomonitoring program in the Nebraska, and to use novel epidemiological methods by using like newborn fetal spot blood for assessing different environmental exposures either at birth or in utero. These should be the future directions, and I think our findings would help policymakers to make informed policy decisions based on these findings. And I would like to acknowledge Buffett Early Childhood Cancer Institute, Dorothy Water for Food Global Institute here at the University of Nebraska, and also the funds that were supported through the Nebraska legislator supporting our research. And would like to acknowledge all my mentors and for guiding me through this study. Thank you.
Thank you so much. It's a compelling set of data for the need to better protect children. So thank you for your work and the continuing work on this. And with that, I'd like to introduce Emile Habimana, who is a doctoral candidate specializing in environmental analytical chemistry at the University of Montreal in Quebec. He holds a Master's of Science in Chemical Engineering, integrated chemical and environmental technology from Hankyon National University in South Korea, a Bachelor of Science of Chemistry from the National University of Rwanda, and a background working in environmental management for the Ministry of Natural Resources in Kigali, Rwanda. Mr. Hamabana's research working with Professor Sébastien Sauvé, PhD, focuses on development of advanced analytical methodologies for profiling and quantifying emerging contaminants within environmental matrices, as well as evaluating the risks they pose to both ecosystems and human health. He's the lead author on a paper published earlier this year entitled A Review of Properties, Occurrence, Fate, and Transportation Mechanism of Contaminants of Emerging Concern in Sewage Sludge, Biosolids, and Soils, Recent Advances and Future Trends. With that, I welcome you, Emile. Thank you for joining us.
Thank you so much, Mr. J, for the introduction. Many thanks to the organization Beyond Pesticide for the invite to speak and share our findings. And today I'm going to talk about the presence, the toxicity, fate, and transportation mechanism of emerging contaminants. So in today's presentation, I'm going to just define what are biosolids, and before talking about their content in fertilizing materials, as well as their contamination potential. After that, I will go through the environmental fate and toxicity of different classes of contaminants found in biosolids, and talk about the potential of mixture toxicity. Before just talking about the recommendation, I'm going to introduce the challenge of analyzing these contaminants in biosolids. So, first of all, what are biosolids? In simple words, biosolids are human manure. Human manure, which are used to improve soil quality, And here I'm just giving a non-exhaustive and approximative composition of them because specific composition depend on each municipality and the wastewater treatment technology. But in general, sewage sludge is composed by more than 90% of water and less than 10% of solid fraction, referred to biosolid after treatment. This solid fraction contain a game of organic matter and inorganic compound with estimate composition percentage here presented. They are very useful because they contain fertilizing materials. They contain both macro and micronutrients. Macronutrients are this kind of element required by crops for their growth and biomass formation. They comprise basically nitrogen, phosphorus, and potassium. And on the other side, we have the micronutrients. Micronutrients are required by crops at low concentration at trace level sometimes, but they are very useful for some physiological and biochemical reaction for growth like photosynthesis, stress tolerance, and so forth. They comprise iron, manganese, zinc, boron, copper, nickel, cobalt, just to name some. All these base nutrients can be found in biosolid, in sewage sludge. That is why in some area they are used to improve the soil quality. On the other side, however, they have been proven to be the reservoir, the sink for emerging contaminants. In our recent review, the one Mr. J talked about, we have reviewed the publication from 2019 to 2024, listing, analyzing the occurrence, the fate and transportation model, as well as the toxicity of these different class of contaminants. And we found out over that time period, 419 contaminants of emerging concern have been studied, out of which 229 compounds were detected at least once across the investigated matrices. As you can see, the percentage, the concentration load is dominated by the phthalates in sewage sludge, which represent more than 97% of compared to other contaminants we have just reviewed. This is a huge concentration, and the previous presenter have talked about their potential as endocrine disruptors and so forth. They are followed by pharmaceutical products, personal care products, hormones, rubber and polymer antioxidant, and bisphenols with the percentage concentration presented in this graph. What you want to highlight here is that really these are very important biosolid or other organic fertilizers are important sink of these contaminants. As well, I have talked about these categories, main categories. I want to say that each category can also be broken down into specific classes and subclasses. Let's talk about the pharmaceutical products. So every medication we take can be found in this human manure. We have found that almost all therapeutical classes of medication are found there. We have just identified 18 classes which can be found in our review, but here I just listing some of them like analgesics, antibiotics, anticonvulsants, anti-anxiety, cardiac care medication in different classes, antifungals, lipid regulators, cancer treatment drugs, and erectile dysfunction agents, just to name some. As you can see, let's also show the example from the analgesics. We have the Tylenol, which is also known as paracetamol or acetaminophen, and the Advil. Both are non-steroid anti-inflammatory drugs. So, but on the other side, we have the codeine and morphine, which are opioid analgesics, which have different mode of action and different biological target site within organism. And this is predicting what mode of action as a mixture toxicity they may have when they are in the environment. I'll come back to this later. But what I want to highlight here is that we have almost everything in this sewage sludge. The same applied to other categories of these emerging contaminants. In personal care product, we have preservatives, antimicrobials, insecticides, fragrance, and UV filters. In hormone, we have just listed natural and synthetic hormones. Excuse, just apology, I have some disturbing outside. So, and we have plasticizers and the plastic-related compounds like phthalic acid esters, also known as phthalates, and bisphenol. We also listed the industrial antioxidants and petroleum-based products and neonicotinoid pesticides. All these are form a cocktail of contaminants in biosolids. Here we listed the top most 30 pharmaceutical products found in sludge and biosolids. We can find miconazole, an antifungic, followed by fluoroquinolone antibiotic or fluoxacin. But what we want to show is that we have a large gamut of pharmaceutical. If we list all compound or contaminants from all the category, we can find almost all classes represented. So we have the phthalates, we have the plasticizers, antioxidant, personal care products, and you can see them. But surprisingly, we are not seeing pesticide, neonicotinoid. We just found in the literature in the most concentrated, most recurring contaminants. And this is very important in respect of what I'm going to talk with regard to the transformation products. The neonicotinoid pesticides which were just covered by different researchers in past 5 years have analyzed 9 pesticide neonics, and 7 of them have been detected, at least positively detected in soil sample analyzed. So as I'm going to talk about the environmental effect and toxicity, I'm going to request your attention when I will come back to the pesticide neonicotinoids, because the transformation product is very key. But before I embark on that, allow me to talk about the bioaccumulation and plant uptake by different category of plasticizers, of pharmaceutical and personal care products. In this study, they assessed how different physicochemical properties of this contaminant will drive their uptake by crops. For pharmaceutical and personal care products were evaluated, and the 2 properties were partition and octanol water partition, which in simple word translate the fat solubility and water solubility of them, have been proven to be the guiding metrics on their behavior and their fate during the bioaccumulation. As you can see, the triclosan, which is fat soluble, will be accumulated in roots. And gymnofibrozil, which is a lipid regulator, also will accumulate in roots. However, fluoxetine, which is water soluble, will be in less quantity in the roots, but it will get increased as it moves up in the plant. Now, the fluoxetine will be accumulated slightly in the roots, but the accumulation will be much found in the stem and less in the leaves. The opposite scenario for carbamazepine, where the higher concentration is found in the leaves. So let's make a scenario where, let's say, you grow different vegetables in a field amended by this biosolid and where these four contaminants are found. It is likely that if you grow carrots, which represent the roots, asparagus, representing the stem, and cabbage, you will find much triclosan and gemfibrozil in carrots, and you are likely to find much fluoxetine in asparagus and much carbamazepine in the cabbage. So this is the deduction you can just assume to learn from this bioaccumulation potential. So what I want to highlight is that individual compound, individual chemical, individual contaminant will present unique fate and transformation and toxicity that is unique to itself. Other presenters have also talked about phthalates, which are used in personal care product and daily use and daily use product and toys, to which humans, both adult and children, can be exposed. And the toxicity has been mentioned. I don't think I need to repeat this. Allow me to talk about also hormone that we have found natural hormones detected in these fertilizing materials, but also synthetic hormones. Here, I want to highlight that synthetic hormones are usually designed to resist to metabolic degradation, whereas they can be more persistent than natural hormone in the environment. This is the case of ethylene estradiol, which is used as a contraceptive pills, whose the persistence has been documented in water and sediments. As I want to highlight the role of transformation product, allow me to start with the hormones. As you can see in this red square, we have the ethinyl estradiol, the one I just introduced as a contraceptive pill. In the environment, it can undergo some microorganism degradation and be converted into other form of hormone. But the most appealing is about the conjugated hormones, because hormones are active, are biological active in their deconjugated form, like the ethinyl estradiol and the three others above. But when in that deconjugated form, they are fat soluble to be able to cross the cell membrane and to reach the biological target site. But when the organism want to get rid of them, it is going to conjugate them with some chemical group like sulfate and glucuronide, and this is what I have in these green boxes. So this conjugated form of hormones, when they are excreted in the environment, in the sewage sludge, they can undergo other transformation and reacquire their deconjugated form and become biologically active. And the challenge we have seen in the literature and the research is that most analytical techniques focus on the deconjugated form, letting aside the conjugated form as if they are not toxic. But as you can see here, in the environment, they can regain their biological active state. That is why it is important as chemists develop analytical method to consider both conjugated and deconjugated forms of hormone to ensure they have a comprehensive data that consider all types of hormones we may have in the environment. So, as I have said, In the 50 most recurring, most prevalent contaminants we have documented in our review, we didn't see any neonic insecticide. But that doesn't mean they shouldn't be there. And I want to use this recent publication on the transformation product to prove that the fact that the parent compound is not there doesn't mean it doesn't pose any exposure to the environment, or its potential toxicity should be excluded. Here we have the example of some of the neonic pesticide. We have the imidacloprid, which can undergo some chemical transformation within the organism or in the environment, and is going to generate other transformation product that may even be uh, more toxic than the parent. Here we have also the acetamiprid. For some of us or some of you who are aware of that, this has raised a petition in France because they want to return the use of acetamiprid, which was banned back in 2018. But there is actually a public petition asking really not to reintroduce this acetamiprid. As other pesticide neonics, you can see it can undergo the transformation within the organism or in the environment. The same for nitenpiram and thiacloprid. All of these neonic pesticide can be metabolized or decomposed in the environment to generate transformation product chemicals that we need really to evaluate. In this study with the Environmental Science Technology Journal, the authors have reviewed the occurrence of the transformation products of neonics and their transformation products. So on the histogram on the left side, we have the parent neonics, and on the right side, we have the histogram for the transformation product. These neonics and transformation products were analyzed in 13 human matrices, including serum, blood, and so forth, as you can see on the list. Surprisingly, the parent neonics were found to be prevalent in human matrices in concentration ranging between 0 and 2 nanogram per milliliter or nanogram per gram. However, for the transformation products, you can find that the concentration can increase up to around 6 nanogram per milliliter or nanogram per gram. This study highlights the importance of analyzing the transformation product of different chemicals we are analyzing in these complex matrices. This take me to the mixture toxicity. In general, there are 3 or 4 models to predict the behavior of different mixture in the environment. When different contaminants are going to the same biological target site, they may have a concentration addition. If they affect different biological target site, they may have independent action. And this will lead to a scenario where the toxicity of mixture is equal to the sum of the individual components. I have talked about this when I introduced the, for instance, the analgesics. Let's say that the Tylenol, the naproxen, the ibuprofen, which may have the same target site, may have an additive toxicity, but that will be different from that of opioid analgesics like morphine and codeine, which are going to target the nervous system. The second scenario, or the third scenario, is where the chemicals, the contaminant will interact between them, having either a potentiation in a situation where the toxicity of mixture will become greater than that of the sum of the components, or we will have a competition at the target site and the toxicity of mixture will be less than that of the individual components. So the question is, what model is more appropriate compared to the others? So this will depend on the type of the contaminant studied, but what I want to highlight is that all the three models are possible in the environment. So there is then, after seeing all this list of contaminants, there is a challenge in developing the analytical technique to analyze them. In my case, I have listed the most recurring contaminant, pharmaceutical, targeting different therapeutic groups, personal care with different uses, hormones, considering both natural and synthetic. And I have the column of the analyte that I have targeted in my method, and plasticizers and plastic-related compound. I also considered the antioxidants, the sweeteners and the cooking byproducts, the pesticides with a focus on neonics and herbicides, and persistent, mobile, and toxic compound. And they have listed a total of 156 compounds for which I want to develop an analytical method. So there is a tedious work with the developing a method, but this is the process we followed in the method we developed. Our article is under review with the Journal of Hazardous Materials, but this is the technical process. I don't think I need to dive into this, but I want to show there is really a work behind of preparing sample, extracting the target analyte from the matrix to ensure that you are getting the analyte and removing the interferences. We have the separation using chromatographic technique and the analysis using the mass spectrometry techniques, which is really the most used, the state-of-the-art application of chemistry in analyzing these contaminants. So, as I said, I targeted more than 150 contaminant, but the method was validated for 74 contaminant. Here, what I want to showcase is the challenge in developing a method. It's not that a method is going to work for all the list of the contaminant you have. And serving at the baseline of the toxicity assessment and the regulation, the analysis of the contaminant we have in our matrices remained really a challenge. But here, what I want to highlight, we have for the first time quantified this nitroimidacloprid, which is a transformation product of neonic imidacloprid in sludge and biosolids. So, as I said, it has never been detected before in fertilizing materials, and in this case, having it in biosolid can show that we need really to expand analysis not only of the parent neonics but also the transformation products.
So, I'm sorry, we're going to have to jump. I know you want to get to your recommendations and acknowledgements. If you could wrap that up in a minute, that'd be great.
All right, thank you. So we recommend using advanced techniques and grouping contaminant as we assess them, and use some technical available to analyze them. And the recommendation we will do, we need really to make a link between what we consume as the medication and to assess them in these fertilizing materials. We need to go after transformation product, but some of the contaminants which are used in the developed world have been found in isolated area like the Arctic, and for them we need really to continue their evaluation. The other element I want to highlight is some accepted daily intake, which represent what is seen as the quantity that someone can consume with acceptable risk, But with the existing one, for instance, in Europe, recently in 2024, a threshold of 0.2 nanogram per kilogram body weight per day for bisphenol A was established. But this was really one of the good acceptable intake, that threshold that was established, but it still lacks some consideration because it is taking into consideration of bisphenol A only without other analogs which has the same toxicity. I think that is what I have to say, and I thank you for your attention and the time. I look forward to receive the question if there are any. Thank you so much.
Thank you, thank you. Wow, what an amazing group of scientists, and your work is so important. We go into so many communities where the park director tells us that they have this great, very inexpensive fertility that they're using, fertilizer that they're using on their properties, and it ends up being biosolid sewage sludge. So if we could have the speakers turn their cameras on so we can see y'all, Carolina and Jabin and Emil. Is Carolina still? Yeah. Thank you again. We can see your work. You know, when we brought you all together for this event, we knew that you had very specific areas of research that we viewed as cutting edge, and you established exposure patterns, and you established health outcomes, adverse effects associated with those exposure patterns. But the other thing you all have done is talked about mechanisms at work, mechanisms both in terms of how these chemicals interact in our bodies, how they interact in the environment, how their properties cause them to fall into categories that have direct effects as well as epigenetic and transgenerational effects. And from that perspective, we see the themes happening here. Let me start with you, Carolina. There is a question about specifics about your findings around how far people were from the actual site of exposure and whether there was any correlation associated with the distance from where the applications were taking place. Obviously, there's drift and so forth. I'm fascinated by the laundry issue, which years ago there was a study in Iowa of farm families where they found a very similar effect. And these sorts of exposure patterns are not picked up in the evaluation of regulators. So they're subtleties. But you're talking about rural women and an occupational exposure, but also exposure associated in the home related to an occupational exposure, which is sort of a secondary exposure. But then you're getting deep into the mechanism around breast cancer, which also is, and again, this is thematic, right? There's a failure to follow things all the way through, follow the impact of an exposure and then the secondary exposure and the impact of that secondary, that being laundry, a breast cancer effect, but the mechanism around that breast cancer effect that with immunological effects and what that means to effectiveness of cures or effectiveness of potency of the tumor. And I know, Brazil, obviously you guys are under a lot of pressure to produce more for export and to get, become part of the world food system in that respect. And we eat a lot of your food here. I'm just wondering how you see as a scientist the ability to apply these complexities to a regulatory framework.
That's an excellent question and a really hard one to answer as well. So here in Brazil, we live in a country as you know, that has recently had a very negationist or deniable administration. The previous administration was against science, and it was really hard to transform scientific knowledge into actions that could benefit the population. But after we identified the problem of women doing laundry and getting contaminated, we started to talk to the current administration, which is much more open to these types of dialogue and the need to transform scientific knowledge into public health policies. So we've been able to talk to municipal managers in the municipalities that were part of the study. So 27 municipalities, and at the state level, the Department of Health, which is the department responsible for vigilance of women farmers. We've also been talking to them, and the Ministry of Women at the federal level, under President Lula, he created a ministry for women issues. So we feel like Brazil is starting to look into that. And there is an incipient attempt to start a conversation based on all of these complex mechanisms. And it's been really hard then to translate all of these findings so that the current administration can understand that science needs to be used as a basis for the decision-making process. But in the current administration, science has been well accepted and has been used as a foundation for many policies. But something that saddens me is that although Brazil held COP30 recently in the Amazon, on the same day we had the approval of dozens of new pesticides that were not even needed. Because, as I said, Brazil is known for having a huge amount of pesticides that are banned elsewhere but allowed here. And the same day, they announced that atrazine would be reclassified as probably carcinogenic. So all of these things happening in the country. On one hand, we were trying to discuss the climate issue. The use of pesticides was actually debated during COP30 as well. And we scientists are struggling here to talk about what's going on. Other colleagues here talked about contamination of children. And there is a study showing that the children of women farmers have more cancer, especially pediatric leukemia. And their response to treatment is also worse. So we are just concluding this study to publish that. So we've been, agriculture is key here for Brazilian agriculture. We are a leader in agricultural exports, but we're actually selling our lives. Whenever we export a product and a few get rich, because the people who are getting rich with agriculture here in Brazil, they do not drink our contaminated water. Our water is contaminated with 27 pesticides, and 11 of them have carcinogenic potential. And people who make money with this don't eat the food that we eat. They live in Europe. They live in countries that have pesticides very well regulated. So it's really hard. We have to be careful when we talk about the application of these findings, unfortunately.
Yeah, there's some similarities, as you probably know, with the United States and under our current administration, where we're seeing deregulation as being a positive, as well as dismantling many of the programs. But for those who haven't heard, the internet, Carolina referred to atrazine and carcinogenicity, and the International Agency for Research on Cancer has made a determination that it is a human carcinogen, or they call it probable human carcinogen, which is the same classification they gave to glyphosate several years ago. Well, thank you for that answer, and you've opened up, a whole conversation that we'd like to continue with you. and, see where there can be collaboration on this. Moving over back to the United States, in Nebraska, in the heartland, right? You're taking on some really big, powerful interests there. I mean, you've got not just the heartland, but you've got agribusiness, powerful interests, and you're raising very serious questions about the impacts on children. Um, I'm wondering also in the context of the politics, do you find that there's any pressure on the medical center to sort of tamp down these sorts of research findings? And we did get a question from Wisconsin by a medical doctor who is wondering whether you're aware of other studies like this out of other medical institutions in other states, especially Midwestern states.
Yeah, thank you for this interesting question, Jay. Yeah, I haven't, like, in the recent time since I have published this, I haven't come across any specific, like, mixture study, like, as such, that is relevant to the pediatric cancer. But yes, there are non-mixture studies, like specifically related to specific pesticides, mostly in the Western states. Most of these studies, case control studies, like coming from California, but yeah, I haven't recently come across anything.
Do you think studies, sorry to interrupt, but do you think studies like this, especially out of the medical school, I know you're sort of in public health, but do you think that it incentivizes any people that you work with or interact with in the medical community to engage more in a public discussion around, because they're treating the cancers, right? It'd be really interesting to know, of the cancers you're finding, how many are actually treated at the University of Nebraska Medical Center, and talk to the doctors that are treating these cancers, these children. And so have you, and I know this is a difficult question. One of our strategic goals as an organization is to engage the medical community, to take what they're treating and engage them with the information that you've generated and others to advocate for a preventive strategy. Can you
Yes.
envision that happening?
Sure, yeah, because I just came back from a Children's Health Research Institute like scientific conference. Yesterday we had this conference. So, there is a lot of emphasis on the pediatric cancer research here within the University of Nebraska, like Medical Center, because yes, I mean, in the state of Nebraska, like UNMC is, and the Children's Nebraska, these are the bigger healthcare systems which are treating the cancer. And it is interesting, and it really fascinates me to see the clinician scientists actually, they are working in this area of drug discovery related to pediatric cancer, and also working with the government here to mitigate more funds for the pediatric cancer research. I see that already happening here.
Yeah, well, that's good. I mean, that's what's going to need to happen to create that bridge and to have this discussion, at a high level within the organizations that represent the medical community. The American Pediatrics Association has been active over the years in evaluating exposure to children, and has even in one of their publications advocated for the support of organic agriculture because they have found lower residues, or some cases no residues associated with that, which is a segue over to Emil. Thank you for that, Shabin. One of the things we fought for really hard in the United States was a definition of organic production that prohibited the use of biosolids, sewage sludge, in the production. So you cannot be certified as an organic producer if you're using sewage sludge or biosolids. And people always ask, why are you guys engaged in issues that don't involve pesticides? You take a pesticide, a crop that's produced, say, and then is processed. And there are all these processing aids that go in. And I served on the National Organic Standards Board in the US, and we had to evaluate all those processing materials, some of which end up in sewage sludge. But we carefully go through this process of determining, what makes something compatible with nature, or what is a process that's ultimately compatible with nature in a way that, yes, it may have positive elements like you indicated with the macro micronutrients, but has a darker side in terms of negative aspects associated with the contaminants. And so we have to begin thinking holistically. That's why I really wanted to have you on the program here, because we have to start looking at whole systems, not just the individual pieces of that system, but step back and look at the whole system. And when EPA registers a pesticide, they look at use. When the U.S. Environmental Protection Agency, and this is true around the world, they'll look at use. They're not looking at, well, what's the disposal issue here? Where's this stuff going to go when it's disposed of, or when it goes down the drain? By the way, you mentioned triclosan there, which is an antimicrobial registered as a pesticide. And also the antibiotics are widely used in agriculture as well. And we're concerned about antibiotic resistance. But again, your finding of the degradation product and the transformation materials, are you seeing a lot of that in the scientific literature? When you looked around in your own work, are you seeing that discussion happening broadly in the scientific community?
Thank you so much, Jay, for your question, which is very interesting. And I love the introduction you make, seeing things holistically. That's number one. Why? Because we are having human society that is going to generate sewage sludge, which is going to increase in the future, which we need to dispose of. That's number one, holistically. I was lastly modeling the sewage sludge, and I've seen that with making an island like Montreal as a dumping site, we will have it filled within 1 year with a depth of 5 meters with all sewage produced in the world. So we need to think holistically and in a circularity model. That's number one. Number two, we agree with you with this load of contaminants. Sewage sludge, biosolid should not be used for production. No, should not be. However, we need to see this by region and local contexts. They hold some toxicants and some soil amendment element which can be used to improve the soil quality for further use, because the contaminant can degrade with the time. And what we're trying to see is at which extent we can advise policymakers in regulating these sludges.
Yeah.
But yes, I agree, they should not be used for production.
Yeah, and there's obviously around the world now we're seeing the contamination with PFAS, which sort of drives the point home because it is so persistent and, as it's called, a forever chemical. Well, we have a lot of forever chemicals as you've discovered in your research. Wow, I could talk to you guys for days. Okay, we got to get back together again and continue this conversation because I think it's all interrelated. I'll leave on this note. We had another question about impacts on the central nervous system and Parkinson's disease, which is a topic we didn't directly address today, and I wasn't going to ask about that. But it was interesting to note that you, Jabin, discovered a pattern of central nervous system cancer, if I read that correctly, CNS cancer. So obviously we know that these chemicals are adversely affecting the central nervous system in all kinds of ways. And that leads us to the relationship with Parkinson's. So we will end on that note, and we cannot thank you enough, not only for being here today, but for the work that you are doing. If we can be of any assistance in any way, please be in touch, and we will be reaching out to you again very soon. Thank you for the time today, and keep up the great work. Take care, and thanks for the participants joining us today. Signing off.